in this interaction of genes, again I'll explain you the definition or the concept. Interaction means interacting two or more than two genes. They come together and they work together for a particular character. Now that work, it may be positive or it may be negative, but they work together. So we say that those genes, group of genes, they interact with each other. As a result, they show a particular character. So that is the interaction of genes. In this chapter, we are going to study the positive as well as negative interactions. In that, whatever dihybrid cross is there, which was explained by Mendel, that a dihybrid cross 9:3:3:1 is modified because of this interaction of genes, and they are modified in these ways. It is modified into 9:7. That interaction is called as complementary gene interaction. It is modified into 15:1. So that interaction is called as duplicate gene interaction. Then it is also modified into 12:3:1. So this type of interaction is called as dominant epistasis. Then it is also modified into 9:3:4 ratio. It is called a recessive epistasis or supplementary gene interaction. Then it is also modified into 13:3 ratio. It is called as inhibitory gene interaction. Here the monohybrid ratio, that is 3:1, is modified into 2:1, and that concept is called as lethal gene interaction. So that lethal gene interaction we are going to study with the example of inheritance of coat color in mice. Then we are also going to study the inheritance of sickle cell anemia. So these are the some of the points that we are going to study in this chapter. So as in last lecture, I told you that generally while studying Mendel's dihybrid process, it was found that two genes control two different characters. Means for example, R gene, it controls the shape of the seed. Y gene, it controls the color of the seed. It is, however, not necessary that a single character is controlled by one gene only. Instead, it may be controlled by more than one gene. In such a situation, when two or more than two genes, they come together to give rise to a particular character or phenotype, that type of interaction is called as interaction of genes. For example, R gene is responsible for shape of the seed, Y gene is responsible for color of the gene, but it may, those two genes, they may come together and they may give a third character. It may not be the shape or may not be the color, it will be the different character. So, for example, what? Suppose there is a gene A which is responsible for giving one color or one character, that we will call it as phenotype A. B is responsible for giving phenotype B. But when this A and B When they come together, they give rise to the third character, that is C. Neither A character is seen nor the B character is seen. We observe the third character, that is phenotype C. So these two genes, when they are dominant over their alleles, they interact with each other and they give one specific character, a new character. We call it as phenotype C. This is possible only when A gene and B genes both, when come together, they are in dominant conditions. Both A and B, if they are present in that particular organism in dominant condition, then and then only we can observe this phenotype C. But if they if any one of them is absent, for example here capital A means dominant A gene is absent, so dominant B gene will be present, then we will not get this phenotype C. We will get phenotype B only because B is dominant here. Here B is absent, dominant B is absent. It means that dominant A may be present. So here also again we will not get phenotype C, but we will get phenotype A because here A is present. When both dominant A and dominant B are absent, means there are alleles, recessive alleles, that is small a and small b are present, then also we will not get this phenotype C. So such type of interaction is called as interaction of genes. Means A and B gene, they come together, they work together, they interact with each other, and they show a particular different phenotype. Let's see those interactions with certain examples. Now, this is the first type of interaction. We are going to see different types of interactions. So this is the first type of interaction. That type of interaction is called as complementary gene interaction. In last lecture, I told you that these interactions, they may be positive or they may be negative. They may be positive or they may be negative. Okay. All of you, are you understanding? If you are understanding, please write in the chat box that you are understanding so that I will also take your attendance from the chat box. All of you. Write in the chat box that you are understanding all those things so that I will take your attendance also. Hurry up. Okay, so we'll start with the first type of interaction, that is complementary gene interaction. Now, as I said you that this interaction is the positive type of interaction. Means what? If two genes are involved in a specific pathway or responsible for producing a particular phenotype, such interaction is called as complementary gene interaction. This type of interaction was first observed by Betson and Punnett when they crossed two white flowered varieties of sweet pea, that is Lathyrus odoratus. These two scientists, Betson and Punnett, they crossed two varieties of sweet pea, Lathyrus odoratus. Both the varieties were giving white flowers. Means the male parent is also producing the white flower, female parent is also producing the white flower only. Okay. So in F1 generation, what should you get? What color of the flower should you get? Please write in the chat box. Now again, listen what I am saying. The male flower is producing white flowers. Female plant is also producing white flowers. Male plant is also producing white flowers. What type of flower should be produced by their progeny in F1 generation? Write in the chat box which flowers should be produced by those parents which are giving both white flowers. If we cross two plants, one is white, another is also white. If we cross them, what will be the next generation producing flowers? What color of the flowers will be produced in the next generation? If both the parents are producing white flowers, what will be the flowers of their progeny? Color of the flower. Yes, hurry up. You are taking so much time. It is not required to take such a big span. Answer is very easy. Yes, Govind, Shubham. Yes, Vaishali, good. So all should produce which color? White. Okay, Divya. All should produce white color flowers. But now listen carefully. But Betson and Punnett found that the F1 progeny, it produced colored flowers. Means it produced purple color flowers. Now listen carefully what I am saying. The male parent is producing white flower, female parent is also producing white flower. So according to Mendel, the F1 generation, F1 progeny should also produce white flowers only. But Betson and Punnett found that F1 was producing colored flowers. Means purple color flowers were produced. So this was a magic. This was a wonder. So how did this happen? For searching how it happened, for searching the reason, what they did, they allowed these F1 plants to self-pollinate for the F2 generation. When F2 progeny was obtained from F1, so plants with colored flowers and those with white flowers were obtained in 9:7 ratio. So this was the modification of 9:3:3:1 ratio. So this is a ratio of dihybrid cross. So here, instead of getting some flowers purple, some flowers white, here they got the flowers in the ratio of 9:7. So 9 were purple, 7 were white. So that is the modification of 9:3:3:1 ratio. Now you can say that, sir, how this 9:3:3:1 ratio is a dihybrid cross. Dihybrid cross means we are considering two characters. Now here only we are considering only one character, and that is the color of the flower. So how you can say that this is the modification of dihybrid cross? So the reason is that if you count 9 plus 7, it becomes 16. If you count 9:3:3:1, it also becomes 16. So it means that both the values are same. 16, here also 16. So this is the modification of this ratio. Now how this happened? The parents were white colored flowers. The progeny F1 hybrid is with purple color flower. And again in F2 generation we get white as well as purple color flowers. Now how this happens? So Betson and Punnett, they explained it with the example. And that example was the Lathyrus odoratus plant. Lathyrus odoratus plant. Okay. So a pea plant with white flowers with genotype capital C, small p is crossed to a plant with white flowers with genotype small c, capital P. Now here suppose this is a male parent and this is a female parent. Here C gene is dominant, P gene is recessive. Here C gene is recessive and P gene is dominant. Here the plant gives white flowers. Here also the plant gives white flowers. It means that capital C is responsible for giving white flowers as it is dominant. Here in this plant, capital P is dominant, so it is responsible for giving white color flowers. In short, we can say that when capital C is present, P is recessive, then we will get white flowers. Here when capital P is dominant and C is recessive, then also we get white flowers. Means dominant P is responsible for white flowers, dominant C is responsible for white flowers only. So what will happen if they come together in F1 generation? So the F1 plant will be capital C, recessive C, dominant P, recessive P. So this will be the heterozygote. Now here dominant C is also present, dominant P is also present. So C was responsible for white flowers, P is also responsible for white flowers. So here we should get white flowers only in F1 generation. But we are getting purple color flowers. When dominant C and dominant P, they come together, they change the color of the flower. They give purple color flower. Instead of giving white flowers, they give purple color flower. So this is the interaction of genes. So capital C, dominant C is interacting with P or P is interacting with C. As a result, we get a new color. So they are working together for a particular character, and that character is not harmful. This purple color flower, it is not harmful to the organism or to that plant or to the nature. So they are working together for a good job. Okay, so purple color, we were getting white color flowers. Now we are getting purple color flowers. So it is a good thing. Okay, So they are working together. So as they are working together, we call it as gene interaction. And as they are working together for a positive purpose, for good purpose, we call it as complementary gene interaction. So the normal ratio from selfing, the dihybrid, is 9 is to 3 is to 3 is to 1. 9 is to 3 is to 3 is to 1. So when we self this F1 hybrids, the F2 generation which we receive is in the ratio of 9 is to 3 is to 3 is to 1. But if we allow these genotypes or these organisms, these F1 progenies, to self-pollinate in the interaction, capital C and capital P genes give a modified 9 is to 7 ratio. Now, how this ratio is obtained? The following table describes the interaction for each genotype and how the ratio occurs. So the explanation of modification of this 9 is to 3 is to 1 ratio is given with this diagram and this table. Okay. Now, there is one precursor inside the flower on which the capital dominant C gene it acts. So this is one raw material. For example, this is some raw material which is present in the petals of the flower. So on that raw material, this gene it acts. Now how it acts? This gene, dominant C gene, it produces one enzyme. That enzyme is called as enzyme C. Gene C, it produces one enzyme called as enzyme C. That enzyme, it acts on this precursor, and after acting, it gives one intermediate. So that precursor, that colorless thing, it is changed to one color. Okay? What is that color? It may be the white color. Colorless, white. So watercolor, now milk color. Okay, so white color. Now how that white color came? It came because this gene C, it produced one enzyme C which acted on this precursor and it created one material that was white color. Okay. Now if only C is present, dominant C, and P is in recessive condition, then we will not get this further reaction. If P is in if P is in recessive condition, this further reaction will not be found. If that plant is having genotype as capital C and small p like this, capital C and small p, if it is present, we will get only white color flowers. Further reaction will not occur. Okay. Second thing, if capital C and capital P is present in the same flower, what will happen then? Capital C is also present, capital P is also present. So C will produce one enzyme called as enzyme C. It will create white color flower. Then gene P, dominant P, will also produce one enzyme that is called as enzyme P. This enzyme P will interact, or it will work on this intermediate white color flower, and it will produce one pigment, and that pigment is called an anthocyanin pigment. So this anthocyanin pigment is nothing but a color, and that color is nothing but the purple color. Okay. So if capital C and capital P are present, the flowers are colored because anthocyanin is produced. Now why? Because the enzymes, enzyme C and enzyme P, they work together. C works on precursor, it produces intermediate. Enzyme P works on this intermediate and it gives color. So it means that these two genes, they are working together to create colored flowers, purple color flowers. Okay. So only capital C is present and small recessive P is present here, then this further reaction will not happen. because this reaction, to achieve this reaction, we need dominant P. But if here recessive P is present, then this reaction will not happen. So we will get only white color flowers. So here we will get only white color flowers, because why? No anthocyanin is pigment is produced, because P gene is not functional. Recessive enzyme is not functional. Which enzyme is functional? Dominant P is functional. So here dominant P is not present, so you will not get the anthocyanin pigment. Okay. Now in third condition, if recessive C is present and capital P is present, what will happen? So if here recessive C gene is there, then you will not get enzyme C, capital dominant C. As a result, you will not get the intermediate. So as there is no intermediate, this capital P cannot work on this precursor. So if there is no work on this precursor, again you will not get, first reaction is not happened, intermediate is not produced, so the pigment will not be produced. Is it clear? So why? Because C enzyme is non-functional. C enzyme जर dominant असेल तर ते functional असते. Recessive आहे, it is not functional. So here it is recessive. So again you will not get purple color flowers because no anthocyanin pigment. You will get white color flowers only. Now what is the last case? Now in last case, here both the genes are recessive. So neither intermediate will be produced nor the anthocyanin pigment will be produced, because enzyme C is not produced, enzyme P is not produced. Why? Here recessive C is present, here recessive P is present. So they are unable to produce the enzymes. If enzymes are not produced, no intermediate, no anthocyanin pigment. As a result, the color of the flower will be white only. In short, what is the conclusion of this slide? When capital C and capital P, means dominant C and dominant P, they are together. They are together. Then and then only you will get purple color flowers. But when any one of them is absent or both are absent, you will not get colored flowers, you will get white flowers only. So when they are present together, they interact with each other. After interacting with each other, they produce one pigment called as anthocyanin. And because of that anthocyanin pigment, you will get purple color flowers. So that ratio is modified into 9:3:3:1. So genotypes with capital C and capital P, you get 9. With capital C and recessive P, you get 3. With recessive C and capital P, you get 3. And with recessive C and P, you get only 1. So that is 9:3:3:1 ratio. Okay, but it is modified now. How it is modified? It is modified into two forms. One colored and others are white. So how many colored flowers here we are getting in F2 generation? 9 flowers we are getting colored, and 3 plus 3 plus 1 means 3 plus 3 6 plus 1 7 flowers we are getting white. So the ratio 9:3:3:1 is modified into 9:7. So this is the modification of Mendel's dihybrid cross. So this is the diagrammatic representation. If capital C is present, it will produce one enzyme. That enzyme, if it works on precursor, that precursor is colorless. It works on it, and after producing that, after acting on that precursor, it produces one intermediate. That intermediate is also colorless, so you can say that it is white in color. If capital P is present, again it will produce one enzyme. That enzyme, it acts on this intermediate, and this colorless or white color is changed into purple color. Why? Because this enzyme acts on it and it creates one pigment called as anthocyanin, and that anthocyanin pigment is responsible for giving purple color flowers. So if capital C is present, capital P is present, you will get purple color flower. If capital P is not present, if your all the genes are in recessive condition, this reaction will not happen. So if this reaction not happens, you will get only colorless flowers, means white color flower. Or capital P is present and C is absent, then this reaction will not happen. So you will get only colorless flowers, means the white color flower only. If both are absent, these both reactions will not happen. So you will get white color flowers only. So the conclusion is that when capital dominant C and P both are present, they cooperate, they work together to produce one product. That product is called as purple color flowers. So they work together. So we call that type of interaction as complementary gene interaction. So here is the modification of 9 is to 3 is to 1 ratio. Again, I will explain it in short. White color flower, white color flower. This white color is due to dominant C. This white color is due to dominant P. When both are crossed together, we get F1 generation. Now, this F1 generation, here capital C and capital P, they have came together. So as they come together, they work with each other. They interact with each other and they give purple color flower because enzyme C and enzyme P are produced, so we will get purple color flowers. When they are allowed to self-pollinate in F2 generation, we get 9 purple flowers and 7 white color flowers. So here these purple colors, they show that these are the genotypes, they are producing purple color flowers, and these are the genotypes which are producing white color flowers. So capital C, small c, capital P, small p. So what will be the gametes? These will be the gametes. Suppose these are the gametes of androecium, these are the gametes of gynoecium. So these gametes, they fuse with each other, male gamete, female gamete. As a result, you get a zygote here. So all those zygotes here, both capital C, capital P is present. Here also, both genes are dominant condition. Here also, here also, here also, here also. So in the 9 types, 9 zygotes, all the genes capital C and capital P, they are in dominant condition. So we will get purple color flowers. Now here only C is dominant, so we will get white color flower. Here also C is dominant, we get white color flower. Here P is dominant, so again we will get white color flower because there will be no reaction. P is dominant, only white color flower. Here both are recessive, no reaction is happening, then also you will get white color flower. Yes, P is dominant, yes, C is dominant. So single dominance they are there. So we will get white. So as a result, you get 9 purple and 7 white color flowers. So this is the modification of Mendel's dihybrid cross, that is 9 is to 3 is to 3 is to 1, and that modification is 9 is to 7. So in exam they may ask you what is the modification of Mendel's dihybrid cross with the help of this complementary gene interaction. They will ask you to explain this ratio, how it comes. So this ratio comes in the following way that I have already explained you. So with this, I will stop here. In the next lecture, we will study the duplicate gene interaction. So I think all of you must have understood this. So please write in the chat box that you have understood it. Yes or no. Hurry up. Only 1 minute is remaining. Hurry up.